Hello.
Yes, we can hear.
Hello.
Hello.
Can you hear me?
Yes, we can.
Can you hear me? Hello?
Yes, yes, we can.
Hello? Can you hear you? Okay, brilliant. We can continue. Okay, thank you. Now, the essence of doing tolerance is, it's very crucial when we're going to be discussing autoimmune diseases in the 4th year. And for you to understand this, it has got to be a lecture where we have foundation. And therefore, its importance is for us to discuss things you may have heard of: rheumatoid arthritis, systemic lupus erythematosus, diseases that are unfortunate, that arise when the body's own immune system attacks its own self-tissue. So it's something that we need to appreciate, at least the foundation, because what we do in the foundation is what we use to treat it or manage it with pharmacotherapy. So tolerance is a state of unresponsiveness specific for a given antigen or set of antigens, and it applies to the individual in that it is a state of unresponsiveness to its own tissues. So it's thus the lack of ability to mount an immune response to epitopes to which an individual has the potential to respond. It is specific. It's a negative immune response. It is induced by prior exposure to that antigen. These are just basic facts that introduce the subject. Essentially, when you're asked to define, just hold on a second. It is accepted to be an active process and in essence, a learning experience for T cells. So, by way of explanation, in the intrauterine life, the body must undergo specific immune evaluation such that it does not respond to self-tissues. Both these processes, or this education process, or the process by which we actually initiate prevention of destruction by immune cells, occurs in the bone marrow and in the thymus. In the thymus, it is for T cells. In the bone marrow, it is for B cells. Upon introduction of the subject immunology, I always tend to tell the students that when you are born, you're going to be exposed to almost 1 billion to 100 million to 1 billion types of different antigens. Therefore, one requires T cell clones and B cell clones that are specific for each and every single one of those offending substances or agents. Therefore, it is crucial for the body to first ensure that it is not going to be under attack subsequently when you are born. So, as it says, self-tolerance prevents the body from eliciting an immune attack against its own tissues. All other forms of tolerance are considered non-self-tolerance. And we'll mention that by just way of explanation, but suffices to say our focus today is on self-tolerance. We want to prevent this phenomena, and we want to educate our T and B cells prior to ex utero life. So, example that we talked about non-self, transplant tolerance could eliminate the many drugs with adverse effects given to suppress immunity post-transplantation. It would be of great value in treatment of autoimmune diseases that we've just mentioned. And it is crucial to highlight something that it'll be important for you clinically, is that there is a very significant population of younger individuals, juveniles, young children developing diabetes. Prior to the time we were working as interns and medical officers, this was potentially dangerous, what's it called, disease process, but we're getting people surviving up to 35, 40 with very, very minimal, what's it called, morbidity and mortality. So it's one of the things you'll have to be tackling very actively in this modern age. So, you can see in the middle of that, immunological tolerance can be extrapolated for use in bone marrow transplants, solid organ transplants, autoimmune diseases, infectious diseases, allergic diseases. So it's got a significant amount of input that we need to look at. But suffices to say, when you use tolerance or non-self tolerance, you're effectively trying to manage these disease processes other than autoimmune diseases and allergic. So we divide tolerance based on anatomically defined sites for ease of understanding and teaching, and also gives you an idea as to how the process occurs. So primarily we have central tolerance that is an in utero phenomena. T cells in the thymus and T cells, B cells in the bone marrow, but ultimately both the T cells arise from the bone marrow. The mechanism is clonal deletion, we will mention that. Of the possible, let's say out of 100 T cells or B cells that are educated or similarly enter the site for education, that is, they are evaluated such that they do not produce self-tolerance, 10 to 15% escape the process and released prematurely into the peripheral circulation. But the body, fortunately, has a mechanism of dealing with them when they escape this education system in the thymus or bone marrow, and at the end of the day, they show up in the peripheral lymphoid tissue where they undergo a similar process using different similar procedure or education, but using different immune processes, Anergy, cell death, and immune deviation, which we will highlight as we go down. Suffices to say is that this particular disease processes where you get autoimmune diseases are diseases of females with a ratio of 10 to 1, tends to occur in fertile female over 40, and previously it had high morbidity and mortality. But nowadays people are living for much longer with many of these afflictions due to increases in developments or scientific developments in management, especially from the point of pharmacotherapy and immunotherapy. Cell tolerance. Tolerance of T or B cells induced during development in the primary lymph organs at the central. The chief mechanism of cell tolerance or T cell tolerance is the deletion of autoreactive T cells in the thymus, so the organism is rendered self-tolerant. Immature T cells migrate from the bone marrow to the thymus, where they encounter peptides derived from endogenous proteins, cell proteins bound to major histocompatibility complex molecules on thymic epithelial cells. So we are targeting cells that react to self-tissue. We refer to them as autoreactive T cells. Now, this is something which is scientific, but it's just the end when you've done ontogenesis of T and B lymphocytes. These are terminal cellular forms of T cells where they actually start generating CD4 plus and CD8 prior to going ahead to finally end up at CD4 or CD8. And they refer to as double positive. They carry both of these cluster of differentiation identification molecules. So double positive CD4 plus and CD8 thymocytes initially undergo random generation of different T-cell receptors. That makes sense. You want to deal with the sheer randomness of the number of endogenous and exogenous peptides that you can potentially be exposed to. So positive selection, also called thymic education, ensures that only clones with TCRs and moderate affinity for self-MHC tissue are allowed to develop. Moderate. Cells whose TCR fail to interact with self-MHC peptides undergo programmed cell death. Those that are too strongly bind, they too also die. So out of this possible 80% or 100, 80 to 85% undergo proper education, the other guys escape. This is just a diagram to show what we've actually talked about. But a negative selection by means of apoptosis occurs when T cells not produce functional TCRs when TCR rearrangement fails. So if for one reason the MHC TCR, what's it called, combination or association is defective or doesn't actually come about, you get negative selection. When T cells have low affinity for the MHC cell peptide complex, or when T cells have extremely high affinity for such complexes. These are crucial to understand, but suffice to say, number 3 is the most important. When the T cells you have actually sent from the bone marrow thymus have extremely high affinity for such complexes. Results in self-tolerance. After negative selection, these cells pass from the thymus in the circulating system. Only 2 to 5% exit as mature T cells. But of these particular, we can call that 100% we referred to. Those 80 to 85% now have to be dealt with. So other mechanisms of self-tolerance, these are thymocytes. These are immature. Thymocytes that express autoreactive T-cell receptors are prevented from maturation. That makes sense because if you want to present something to an incoming T-cell and you are sickle and you're autoreactive, that means the thymocyte, the cell that's going to present, we destroy you. Well, Anergy, autoreactive cells are inactivated, and we'll talk about that mechanism in depth rather than deleted. That means they are actually rendered incapable of autoreactivity. Total editing, autoreactive cells are given a second or third chance to rearrange a TCR gene to produce an appropriate TCR for presentation. And finally, among these, the most important is total deletion is probably the most common mechanism responsible for thymic negative selection, induction of apoptosis. Two factors contribute to peripheral tolerance. Not all self-antigens expressed in the central lymph organs when negative selection occurs, and there's a threshold requirement for finitude to self-antigens before clonal deletion is triggered. Suffice to say is that sometimes you might find odd things at the periphery that were not expressed there. And there are some things, if you notice, when you're developing tissue systems and go back to biology, there are some tissues that develop way ahead of the time before thymic education occurs. And that is the essence of the big problem that we'll highlight towards the end. The fact that there's some tissue antigens that are not expressed maximally in the developing embryo that should have been processed by having the right clone available of T cells to react to that self-antigen that is yet to have been produced or may have been produced prematurely before the thymic education. Therefore, it is a process by which mature self-reactive lymphocytes that recognize self-antigens in peripheral tissues are inactivated, killed, or suppressed. And it is very crucial to understand they can be inactivated, rendered useless, killed, deleted completely, or their functions suppressed. And those are the three particular key issues that are the ones that are going to be expressed in our teaching now, because it's going to explain the mechanism of peripheral tolerance. Okay, let's look at this basically. That's okay. It's just still the thymus. It's okay. Sequestration. Now, what does it mean to be sequestered? Sequestration. When someone wants to have to be sequestered somewhere, what do they want to do? Anybody? When a group of people are sequestered in a hall, they are put in the hall and kept there. Sequestration is actually a way of saying that you want to keep a certain set of things hidden somewhere where they cannot actually be seen or recognized. And this is crucial because there's a clinical application. There are two ways this can happen. And if you let me go back to the front, so there's continuity. Self-antigen may be sequestered or hidden in some tissues and will never be available to T cells. Allows these antigens to avoid encounter with reactive lymphocytes under novel circumstances. The first way is a physical barrier, location of antigen in privileged sites. Cells ignore self-antigens if they're expressed in immunologically privileged sites, the brain, the anterior chamber, the lens of the eye, testis, placenta, and fetus. All right. Now, what is the crucial aspect of this thing? How does it apply to us medically? If you have damage to paired tissues like the eye, testes, these are hitherto uninvolved in the so-called thymic education process. But once the blood-brain barrier, or the barrier of the brain, blood tissue barrier is destroyed, you expose those hitherto unexposed tissues to T lymphocytes. So destruction of one testis can actually result in destruction of the other if the injury is severe enough to expose testicular tissue to the immune system. So whenever you have injury of your lens or your eye, your anterior chamber, your testes, etc., and you get one organ going down or in a paired organ system, please make sure that you actually be aware that you're going to have a immune reaction against the other normal tissue. So it is very crucial that you, in the back of your mind, when you're dealing with any kinds of pathology involving these two, especially eyes and testes, that you are actually in the back of your mind, you're remembering that these are immunologically privileged sites have never been exposed to be educated to not to induce self-tolerance. Then we have the immunological barrier. These have never been processed by functional APCs. Yeah, plasma tissues are most likely to survive in these sites because of the tight control of pro-inflammatory lymphocytes. In these sites, pro-inflammatory lymphocytes are controlled by apoptosis and cytokine secretion. That's nothing bad. Let's continue when we come to apoptosis, which is important. Yes. Now, we talked about inactivation, killing off, or suppressing autoreactive T lymphocytes in the prior slide. But what really happens at the mechanism of the peripheral thing when you have apoptosis and what it actually entails? So, when we were talking about, and it should have been stressed that we initiate an immune process, especially when you have T lymphocytes, the secretion of interleukin-2 mobilizes either other T lymphocytes And when you want to bring that particular immune response to a conclusion or to an end, you actually want to introduce death receptors on the T cells that will cause apoptosis. All right. Now, activation-induced cell death, deletion of cells with high ability for antigen, cells that are persistently stimulated will undergo AICD through the generation of the fast ligand, which is CD95 ligand, which is a death receptor. These are expressed in such cells. They actually, on the cell surface of these autoreactive T lymphocytes, they have signal transduction that actually allows them to undergo apoptosis. Okay, then we also have programmed cell death, deletion of cells with immune response is no longer required. That is a standard response that we talked about. So if you want to bring an immune response to an end, you have programmed cell death, apoptosis, through the normal fast ligand. But if you want to actually engage in an autoreactive, highly active autoreactive T lymphocyte, then you go for AICD, and you end up with destruction of that autoreactive T cell. Anergy. T lymphocytes require two signals to become activated, to proliferate and to differentiate. And that I'm sure must have been mentioned. You need activation, proliferation, differentiation after two signals. So the first is the recognition of an appropriate MHC antigen complex with the RT receptors, TCRs, of the responsive lymphocyte. What it really means, when you have a T lymphocyte engaging or being engaged by an APC, antigen presenting cell, when the two cells meet, there is some degree of so-called three-dimensional recognition between the TCL receptor and a similar biochemical configuration on the APC. But in order to cement or induce a reaction or immune or response from the T lymphocyte, you require co-stimulation and co-stimulatory molecules. So you must have co-stimulating molecules on the APC that have receptors on the autoreactive T lymphocyte. So what happens is we negate co-stimulation. That means we prevent the interaction between the co-stimulatory molecule on the APC and its ligand on the autoreactive T lymphocyte, and reduce a state of anergy, that is unresponsiveness to the antigen, even after subsequent repeated challenges. The cell is there, it functions biochemically, physiologically, but has no immune response. So you have induced anergy, a state of unresponsiveness. And finally, what is crucial here, the role of regulatory T cells. As you read further and as you go through your training in medicine, you'll find out that this is a cell that's attracting a lot of attention, not only in the subject of immunology, but also in terms of immunotherapy. So, T regulatory cells or TREGs or Th3 subclones are also called suppressor T cells previously, suppress the activation of clone-specific T cell activity by secretion of cytokines. T regulatory cells recognize specific self-antigens and sometimes foreign antigens, granted, but they downregulate immune responses or processes when they engage with these antigens in the periphery. And they do that by secreting suppressing cytokines. At this juncture, we take down a small question that could be asked. Maybe, I don't know if we have the 100 MCQ still working. I may not be with you guys, but whatever the case may be, for purposes of reading, I would like to, if you ask a short answer question, you start by asking, what is meant by tolerance? Can you write that down for terms of in purposes of revision? What is meant by the term tolerance? Next, differentiate between central and peripheral tolerance. And the central and peripheral tolerance, discuss the immune processes that lead to the induction of self-tolerance. Under the heading Central and Peripheral Tolerance, discuss the immune processes or mechanisms or phenomena that actually will produce or induce self-tolerance. Leave that alone, that's a bit advanced. Yes. This is just a way by way of mentioning something additional which is put in at this junction, just to have an idea. Tregs account for 10-15% of CD4 plus T cells and express CD25 and alpha chain for IL-2. Tregs are antigen specific and they regulate peripheral tolerance by producing suppressor cytokines such as interleukin-10 and beta transforming growth factor. Now, they depend on continuous antigen exposure to stay active, which is quite important. And all of the antigen reduces the quantities of cells. In other words, it is antigen dose dependent in its expression. Okay, this is fine. This is reducing induction about tolerance of various kinds. I won't want to worry about this one. Yeah. Good. The application of tolerance. This way, I hope you have done. Have you done tumor immunology? Hello? Hello? Have you done tumor immunology?
Hello.
Anybody?
No, we haven't.
Hello, Tracy? Have you done tumor immunology?
No, we have not.
Hello? Hello? Yeah?
We haven't done it. We haven't done it yet.
You're not done, but are you hearing me? Yes, we can hear you. Okay, we're not done it. This is crucial. So I won't mention this now. I'll get guys confused. Huh? Just leave it for fourth year. Yeah, okay, fine. All right. Okay, fine. That is the case. We shall put this for fourth year. It will be discussed in depth, but nonetheless, this is an important slide if you want to look at it in terms of being a bit ahead of the class. It is the central area that we're working on cancer therapy, especially in breast, renal, melanoma. So apart from this, that should be enough to survive any exam. So on behalf of myself, I will be now going into a period of sick leave lasting 2 to 6 months. We may or may not meet again, but whatever the case might be, best of luck. So I'm processing my papers for either a 2-month clinical leave or 6 months. I got a couple of things sorted out with myself in terms of the obesity, hypertension, metabolic syndrome, etc. So if I should not see you or not hear from you, best of luck in your career. And hopefully we can meet from place to place. But I don't want to say a premature goodbye. It's just to say it's ciao, but not goodbye completely. I'll be around, but most likely I will not be in the campus past December. So on behalf of myself and last 20 years, best of luck and take care of yourselves. Bye.